What's
Hey
up?
man, hello.
How's it going?
Doing all right.
Uh-huh.
The usual. Hold on. Let me request to record right
Yep.
away. All right, cool. It's recording.
Mm-hmm.
Okay. Honestly, let's just get straight into it.
Yep.
We've spoken about everything that needs to be spoken about. Okay. Alrighty. Um, okay. So this was the first passage. Want to go over 1 second, go into it. Okay. Yeah.
Okay. Now, is this something like you want to go over the passage, or like is it specific questions or what?
So I think we might as well go over the whole passage just because
Let's do it.
I got the whole passage wrong.
Mhm.
So might as well, yeah.
All right. So yep, why don't you
Yeah.
start off by starting to read.
Yep. Okay. The natural development and repair of bone tissue provides a good example for the design of mineralized tissue fabrication schemes. Besides cells, bone tissue consists of both inorganic and organic components. The inorganic component is a carbonated calcium deficient nanocrystalline hydroxyapatite. The organic component is a matrix composed of type 1 collagen and non-collagenous proteins.
Okay.
Um, Researchers in tissue engineering have developed a peptide amphiphile that can be used to make a nanostructured fibrous scaffold similar to an extracellular matrix found in bone.
All right. So when you see like, molecular structures, you want to be identifying functional groups. Um, so what functional groups do you see here?
Right. So let's I mean there's like peptide bonds.
All right, show me
Um
where the peptide bonds would be.
so like
Yep.
it's between the carbonyl and it's like an amide bond.
Exactly. Okay, good. All right, what else do you see?
So there's the amino acid side chains.
Yep.
Like so there's like the thiol group cysteine, right? There's so glycine, and then arginine, asparagine. Wait, no, arginine. Arginine.
So
Yeah.
so when you see the thiol groups, what do you think might happen with them, possibly?
I mean, when I hear the word thiol right away, I hear disulfide bonds.
Yep.
Like,
Exactly.
yeah. So that's a possibility.
Yep. All right, what else do you see?
Um, there's a carboxylic, like there's a
Yep.
glutam, or no, aspartate. One thing I was confused about. So on the very left, right, you have the amide bond, and then there's just like a long carbon. Like, does that represent something?
Hmm. Yeah. So notice how they said amphiphile here.
Okay.
So, what the amph part means like both, like, whatever, if you're ambidextrous or whatever. So, here it means that it would be soluble. It's both hydrophilic and and lipophilic.
Hmm.
So, like this long carbon tail or whatever is the hydrophobic portion.
Hmm.
So yeah, whenever you see like a long chain, long like alkane chain or whatever, like because like if you look at a molecule of like a fat, they have those long
Good.
tails. That's the part that is hydrophobic.
Okay.
So there's one more thing there here.
Mm. The
Yeah, yeah,
phosphate,
yeah. So what can that indicate?
a negative charge.
Uh-huh. And probably that because it seems like these are all, amino acids. So what amino acids could this have been before it was phosphorylated?
It was serine.
Yeah, like serine, threonine, something like that. Okay, perfect. All right, you can continue.
Compound 1 contained 5 regions, hydrophobic region, residues that could be cross-linked for additional structural integrity, spacer residues, a residue that was phosphorylated for future attachment nanocrystals, and residues with affinity for cells.
Okay, so they actually tell us there what
Uh-huh.
we just talked about. Um, so yeah, if at the time you weren't sure about what that meant, when they said hydrophobic region, none of this other stuff seems like very hydrophobic, right?
Yep.
So that should be what that means. But yep.
What does affinity for cells mean? Residues with
Yeah.
affinity for cells.
I guess it likes cells, but I can't tell
Okay.
anything else about
No
that.
worries. Yeah. Okay. Molecules of compound 1 spontaneously self-assemble into cylindrical micelles in water at pH 4, but freely dissolve at pH 8. Oxidation of self-assembled micellar compound 1 with iodine resulted in a solid material compound 2 that was stable to alkaline solutions. Equation 2. Reaction of compound 2 with diethyl compound 3 resulted in formation of compound 1.
So all right, so we see compound one at a pH of 8 turns into they spontaneously okay. So compound one spontaneously turns into micelles in water at pH of 4. So that's why we see for equation 1 that the right side, the micelle side, pH of 4, um, and the left side, pH of 8, where they freely dissolve, right?
Mm-hmm.
And then what do we see for equation 2?
I mean, equation 2 becomes solid.
Mm-hmm.
Like a, yeah, I don't really know what else
Uh-huh.
to
And
say.
they said uh that it's oxidation, right?
Okay.
So I'm thinking, well, all right, yeah, fine. We can leave it like that. And then the last thing, the equation 3, seems like, we're adding compound 2, we're adding a dithiol, and it reformed compound 1 and gave this compound 4 thing. Um, what that compound 4 thing kind of looks like compound something. Does it look like any of that? Anything
Like
on the reactant side?
the left side, like compound 3.
Uh-huh. So
It's disulfide bonds.
yeah, right.
Yeah.
Okay, cool. Now, yep, continue.
Researchers wanted to mimic the alignment that occurs between crystals of HA, collagen fibrils, bone tissue, created a peptide amphiphile that assembles into cylindrical micelles as shown in figure 2. Subsequent capture of nanocrystals of HA, followed by mineralization, was used to form synthetic bone. Okay.
All right. So it seems like we have this N here, meaning like this is like one molecule of it or whatever, or like you have multiple molecules and it ultimately forms this cylindrical micelle. So cool. Okay. Good job. So yep. Yeah. Tell me about this question here.
Compared to micellar compound 1, yeah, compound 2 is more rigid as a result of what type of interaction? Okay. So, yeah, I mean, compound 1 to compound 2 it says oxidation. of compound 1.
Mhm.
So I mean, I would think it would be something to do with hydrogen bonding. Something that's oxidized.
So what type of gels exist?
Silica gel?
I mean, like there's native, there's
Oh, yeah, native gel, like denaturing gel.
uh-huh.
Um, mm, yeah, I forgot the reducing.
Reducing, right?
Yeah,
What does that do?
reducing gel. Um, I don't. Does it make it all negative? It makes them all negative.
So um there's it has to do with some of the stuff we've been, well, I guess we've talked about different stuff, but so it would break disulfide bonds, right?
Okay.
Now, when I was asking you about this here, and I kind of like talked a little or kind of like hinted here that they're talking about oxidation. So if we know that reducing breaks disulfide bonds, oxidizing should make disulfide bonds, right?
Okay, so then yeah, it's gotta be disulfide bonds are like the disulfide bonds are covalent, and so it's intermolecular covalent bonding, right?
Yeah, that's the part where it gets tricky because like pretty much every time like you see a covalent bond, it's intramolecular. It's within the molecule itself. Like typically like a bond that we draw as like a straight line is a covalent bond. Like hydrogen bond, like intramolecular forces like hydrogen bonding and all that stuff, we usually draw as like a dotted line or something, right? So so why isn't this intramolecular covalent bonding? So this is like a bit of an exception in this case. It's because like this whole thing we consider to be one molecule. So if we have like, another one of these bound to each other, it will be between the molecules rather than within. So that's why it'll be intermolecular covalent bonding.
Okay. So like the sulfur would be kind of like reacting with the other sulfur of the other compound. Not reacting, but
Yep. So
like
yeah, I would just do it like this. So like, okay, oxidation. So they were talking about, making disulfide bonds which are covalent. So let's get rid of A and C because we know it must be covalent. And then that's where the tricky part happens because typically covalent are intramolecular, but in this case, since they're telling us that compound one, like multiple molecules of compound one, like come together. That's why this time it'll be intermolecular since it's between molecules of compound one rather than within the molecule of compound one.
hmm okay.
Yep.
Um, which amino acid residues were incorporated into compound 1 to promote the adhesion of cells on scaffold surfaces.
All right. So yeah, that time when you asked me about the affinity for cells, looks like they're asking us about it. So okay,
Yeah.
let's see. Okay, so what are your thoughts?
Yeah, so honestly, like I wasn't even sure here because I remember this passage I stayed at the very last minute
Okay.
And so when I was going through, it was just like super quick. And so like I knew there was cysteine, I knew there was glycine,
Mm-hmm.
and I knew that there was arginine.
Mm-hmm.
And like the way I literally did it is I'm pretty sure I just like chose what was there. Like I wasn't sure between A and B, but I just think I just chose like B. Um, I'm not sure here. Like,
So okay. So when we did our when we looked at compound one to look at the functional groups and stuff, and then after they told us more information about those functional groups, let's see if they actually line up, right? So hydrophobic region is here. Okay, that lines up. Residues that could be cross-linked. What does that mean?
cross-link, no idea. Structural integrity, maybe because the glycine is for flexibility of the protein. That's all I know.
So, what do you think could be the cross-link?
Um,
Like it.
like
Yep.
react with one another. Not react,
Yep.
but
Yep.
like
Yep.
yeah.
So, uh, so let's say we had two of these compounds and we connected them together.
Okay.
That could be through the disulfide bonds, right?
Okay, got it.
So, okay, cross-link disulfide bonds. All right, we have a bunch of those. Okay, that seems to line up, right? Spacer residues, which I'm assuming means nothing, like not nothing, but nothing special. Uh, this stuff seems to be having nothing special, right?
Yep.
And then a residue that was phosphorylated. Oh, that's right there, right?
Wow. It's like,
Yeah.
wow. It's
And then
beautiful.
yep.
It's right there.
Yeah, yeah. The and residues with affinities for cells. I don't know what that means, but these are the things that are left.
Yeah,
So that would probably be the best.
that's crazy. It's okay. It's D. Yeah. Okay. I mean, that's crazy. That's the only way really about going about this, right? You just have to sort it out one by one.
Uh yeah, so I mean here, I did not know if it's gonna be in order like that. So that's why, but yeah, then we checked it and yeah, it makes sense. But yep.
Okay. Um, which amino acid was incorporated into compound one? The future site covalent attachment to HA prior to mineralization. Okay. So they're asking about the phosphate, right?
Yeah, yeah. So they also told us here that it was for this purpose, right?
Oh, okay.
So
Serine. Okay. Um, compound one was designed to exhibit pH-dependent self-assembly. Which feature of the molecule is responsible for pH dependence of aggregation? Right. So here, like I just was pretty lost.
yeah.
First to go over aggregation. I kind of forgot what it really was. Is that when the protein folds because of like the hydrophilic is on the outside and it aggregates? Is that what it's alluding to, or
So this isn't even like something that we're expected to know. Um, but they're telling us. So yeah, it's not going to be like the folding stuff. Like so I guess two things. Well, actually one thing. So let's just see. So they're saying that so that there's aggregation happening in equation one. And so if we look at equation one, what happens there?
I mean, like acid base stuff, pH stuff.
Sure.
Don't tell me it's C. Is the answer C?
So I don't know yet, but what is happening here? Like they describe here, right?
Uh, right.
So
I mean, they're responding to pH.
uh-huh.
Yeah.
So um so okay. Molecules of compound one, multiple molecules of that compound one that we have in figure one, like come together to form the micelles, right?
Correct.
So aggregate would mean that like multiple things, molecules add up, right?
Okay.
So yeah. So if you when you see this question, like don't worry about like I forgot or I don't know what they mean by aggregation. Just like look at like the equation that they give us, and then what it would have to mean that they would add up, right? They'd kind of yep, like
A-
ag
Aggregation here means that um a bunch of compound ones are coming together.
yep, to turn into the myself form of compound one.
Okay.
And that's also like that picture all the way at the bottom. Yeah.
Yeah.
So that's what they're yeah, so that's what they're asking. And they're asking what is responsible for this aggregation. Um, right. And like you said, it was the pH, right?
Yeah.
Mm-hmm.
Okay.
Yep.
Okay. All right. So next is this short passage. 40-year-old patient developed a constant thirst, frequent need to urinate. The attending physician suspected the patient had diabetes because, in addition to these symptoms, the patient was overweight and had a family history of diabetes, which both are risk factors for the disease. Diabetes mellitus is a condition in which the body cannot produce or is unable to effectively respond to insulin, peptide hormone made in pancreatic B cells. Normally, insulin promotes facilitated diffusion of glucose into cells from the bloodstream by inducing the translocation of glucose transporter proteins to the plasma membrane. Also helps to regulate the metabolism of fatty and amino acids. Diabetes, the ability of many types of cells to take up glucose is compromised, leading to elevated blood glucose levels. Many tissues then metabolize fatty acids as an alternative energy source, which can lead to metabolic acidosis. Condition of low blood pH. Diabetes can cause protein degradation, resulting in the release of excess amino acids that can be used as substrates for gluconeogenesis, which further increases blood glucose levels. There are two main types of diabetes. So type 1 diabetes, unable to produce insulin, B cells. Okay. While the body often produces normal to elevated amounts of insulin, type 2 diabetes, ability to respond to insulin is compromised. Okay. All right. Glucose transporter proteins in the liver do not require the presence of insulin to facilitate the uptake of glucose. However, insulin does simulate the first step in the glycolytic pathway within the liver. In the liver cells, insulin most likely.
Okay. So from the passage, uh, when you read like up to like from here, so like okay, peptide hormone. I talked to you about buzzwords before, right? Or no?
Yeah.
Okay. So peptide hormone, you know, unlike a steroid hormone, can't just freely travel or pass through the cell membrane, but needs transporters. And they're made in these pancreatic beta cells, and then they're saying that insulin promotes facilitated diffusion of glucose into cells by inducing the translocation of the glucose transporter proteins to the plasma membrane. And then they talk about issues that can cause, like in this third paragraph, issues that can happen from using different things for energy, like fat or protein. So here they're saying that in the liver, the glucose transporter proteins do not require the presence of insulin to facilitate the uptake of glucose. However, insulin does stimulates the first step of the glycolytic pathway within the liver. Therefore, liver cells, insulin most likely. So they're asking, how does, if the glucose transporter proteins don't require insulin to take up glucose, but insulin stimulates that first step of glycolysis in the liver, I guess then what does insulin do, right? So what are your thoughts?
Glycolytic pathway is glycolysis, right? Just making sure.
Uh-huh.
Right, yeah. Okay.
So there's going to be two that we can eliminate right away based on passage information.
C and D.
Yeah, why?
Well, the thing is, the way I thought about it when I actually did eliminate. I got this wrong, but I eliminated C and D. So is glucose uptake into the cell? Is that passive transport?
So they're telling us here facilitated diffusion. So
Right.
it's just so yes, it is passive transport, but it's like through something that helps it.
Oh, okay.
Yep.
Facilitated diffusion. Um, that's you don't use ATP for it.
Oh, correct. Yeah. So anything with the word diffusion in it is going to be, passive, like high to low concentration. So if it so there's diffusion normal and then facilitated, which is just with something helping it to diffuse. But if it uses ATP and stuff, it'll be called active transport.
All right.
So, all right, cool. We can get rid of these.
Yep.
And now, let's see. Hinders glucose uptake by increasing the cellular concentration of glucose. And the aids glucose uptake by decreasing the cellular concentration of glucose. So, what are your thoughts here?
So, they say insulin is not used to facilitate the uptake of glucose.
Okay.
So I was just a little confused because isn't A wrong?
Well, why? tell me your thoughts about
Because if you're increasing cellular concentration of glucose, you're increasing glucose uptake, no?
that. Well, not necessarily, I guess, because so this can possibly help us out.
I mean, glycolysis, yeah.
Uh-huh.
I guess it's increasing cellular concentration.
So what happens in glycolysis?
Oh wait, no, it
Yeah.
removes glucose. Sorry,
Yep.
decreases cellular concentration of glucose.
Yep.
Okay, yeah. I mean, it's B,
Mm-hmm.
but how does that like aid glucose? Like because they say insulin doesn't require insulin to uptake glucose. Insulin most likely, but it does facilitate the uptake of glucose.
So they're just saying here that the glucose transporter proteins in the liver do not require insulin to facilitate the uptake of glucose. Which means that it can do it without insulin, but it doesn't mean that they're saying that insulin does not do that.
Okay.
It's you know how it is on the MCAT, right? It's always like the wording and stuff,
Right.
but you were good in that you were able to eliminate C and D, and then for A and B, and also for like when you have like two different things to look at, like when I say two different things, I really mean like hinders versus aids, and then increasing cellular concentration of glucose versus decreasing. That helps us because, if we don't know, like, if it hinders or aids, we can figure it out if we know, this part.
Yeah. Yeah.
So, yep, if it stimulates glycolysis and glycolysis breaks down glucose, meaning less glucose, um, it's like Le Chatelier almost. Like almost so many things are Le Chatelier.
Yeah.
You
Yeah.
don't have enough glucose, so glucose uptake gets increased. Yep.
Right. Okay. So exercise promotes insulin independent uptake of glucose and working skeletal muscles. Given this regular exercise, most likely reduce blood glucose levels in patients with which type of diabetes. Okay. So when you exercise, insulin gets uptaken independently.
Okay. You mean glucose gets taken in?
Glucose, yeah.
Okay.
Yeah. So I thought like that it would be type 2 because like in type 2, you can't really respond to insulin. And so exercise would promote like even if you don't have insulin, you would have uptake of the skeletal muscles, like glucose uptake in skeletal muscles, right?
Okay.
So yeah, I thought it was type 2.
So okay, so saying exercise promotes the insulin independent uptake of glucose in working skeletal muscles. So like, okay, so the type 2 makes sense, but why wouldn't type 2 also work?
Not sure.
Mm-hmm. So yeah, they're saying that so I get um so they're saying exercise promotes the uptake of glucose. So like if you word it differently, right? Like because so if it's saying it's insulin independent uptake of glucose means that the uptake of glucose can happen whether you have insulin or not. But that doesn't mean that it only happens when you don't have insulin.
Can you repeat that?
So, so if it's saying that exercise helps this glucose uptake happen without needing insulin,
Yeah.
it doesn't mean that it doesn't happen if you do have insulin.
Mm-hmm.
It just means that you don't need insulin for it to happen.
Mm-hmm.
Right? So so it's kind of similar in wording to like the previous problem. Um, you know, if it's saying that it's insulin independent, that means that it can work for type 2 diabetes, but it doesn't mean that it won't work for type 1 diabetes, because insulin independent means that it doesn't need insulin, but that doesn't mean that if you do have insulin that it won't work.
Okay.
So
I mean, so you're saying what's for both?
uh-huh. Because this is saying the same thing as if this wasn't there. Like, it's basically just saying exercise causes glucose uptake. Like in general, right? And that it does not require insulin for that. So there's, so we're basically saying that exercise helps glucose uptake. No, whether you have insulin or not. So if you don't have insulin, you'll have that uptake. If you do have insulin, you'll still have that uptake because the insulin doesn't matter.
Okay.
Yeah.
During the production of insulin, translated polypeptide is cleaved into mature form, secreted from the cell. Cleavage most likely takes place in which of the following locations? Okay. Translated polypeptide. I mean, it's probably ribosome or cytoplasm. So yeah, probably ribosome.
So let's see. So they're asking like where something gets cleaved. And like ribosomes, like compared, so what makes that different from like the nucleus, the endomembrane system, the cytoplasm? Like we never talked about anything happening inside of ribosomes, right?
What is the endomembrane system exactly?
Oh, it's just like the, I guess, the like outside of the nuclear envelope, you get like the endoplasmic reticulum, and then Golgi, like around that, and that's like that endomembrane system. So basically the membranes within the cell, that's the endo part. But the nucleus, the endomembrane system, the cytoplasm are all like, I don't want to say hollow, but there could be things inside of them. Whereas ribosomes are not hollow. They're kind of like solid. So like if it says that it takes place in something, it can't be ribosomes, right? Because they're not hollow.
Interesting.
Yeah, like because another way, like what's the difference like in between like a prokaryote and a eukaryote?
I mean, like prokaryotes don't have the organelles.
Oh, so prokaryotes won't have membrane-bound organelles,
Membrane bound. Okay.
right? Because out of these four, which do they have?
Uh, and they have a cytoplasm.
Oh yeah,
Probably ribosomes.
yeah, ribosomes. So they don't.
Right. Okay.
So yep. So all right, we can eliminate ribosomes because nothing happens inside of it. It's like a solid thing. Um. So okay, now what else could we eliminate?
And definitely nucleus.
Yep. Now between these two, you remember like the signal hypothesis, the signal peptide thing?
Maybe. I don't think so. Like how it travels,
Huh?
like a peptide, like from nucleus to the ribosome to the.
So like sometimes during the translation of a protein, there's a specific sequence called a signal sequence, and that tells the ribosomes that are translating it to pause for a sec. We need to do something a little different, and then it goes to like the rough ER, attaches itself to the rough ER, and it resumes translating the protein inside of that ER. And I'll get a picture of it. So in the chat, I put a picture in the chat.
Okay.
So you could see in that in this picture that once this signal peptide is recognized, the ribosome attaches to the endoplasmic reticulum, and then it continues that translation of that protein inside of that endoplasmic reticulum.
Mm-hmm.
And in there, after it gets formed, like it can there could be lots of like post-translational modifications. So yeah. So this cleavage is going to be happening inside of the endoplasmic like system, because like after the endoplasmic reticulum it can go to like the Golgi, and then the Golgi decides if it's going to be like secreted out like as vesicles or part of like the membrane and stuff. So essentially what we are looking at here is post-translational modifications.
Mm-hmm.
Mm-hmm.
But do they all take place in the endomembrane system or
When you say all,
post-translational
yeah,
processes? They take place in the endomembrane system or the
yep.
or what?
Mm-hmm.
Okay.
Yeah. So here they're telling us that we already have this translated polypeptide, but we must do something to it. It doesn't really matter if it's cleaved or whatever, but if it says that we must do something to it to turn it into the mature form, right?
Yeah.
There must be between the translated polypeptide and the mature form some type of modification, right?
Hmm.
So we have a translated polypeptide, and we must do something to turn it into its mature form, means that we must modify it after it's been translated. And that happens in that endomembrane system.
Yeah.
Yep. So it's a post-translational modification, meaning after translation.
Hmm.
Yep.
Okay. Despite the effects of diabetes, the brain of diabetic patients still receive adequate nourishment. Yeah, I know. I put this. Yeah, I'm pretty sure it was B. Yeah. Yeah.
You get this one.
I got this one right. I got this one right.
All right, cool.
Um, yeah. And this I got right too, but we'll go through real quick. Based on the passage which belongs least likely symptom of diabetes mellitus. So it would have sweet tasting urine, right? Because there's a lot of glucose in the urine,
Yep.
like a buildup of glucose. Um, so unexplained weight loss is due to the fact that it uses up fatty acids, right?
Yeah, as well as like, protein degradation.
Okay. Now, feelings of fatigue. Um, was that because of like constant thirst?
So I guess this is more of like, um, maybe like an inference or whatever that we have to make because it's not really explicitly saying anything like that. But I think what they're saying is that if we're breaking down all of, we're breaking down our muscle and, fat and stuff, but primarily muscle, like you're probably going to be feeling fatigue for that from that.
Because I was just not sure. I like that's the way I got the answer. I basically eliminated all three,
Mhm.
but I wasn't sure like what's the reason for this.
Yeah, it's a little weird.
But
They talk about overweight patients, so I could see how, you know, someone would input that. But I guess they didn't say anything about uh oh wait a second. Sorry, it says least likely.
yeah, 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 yeah.
So um yep. So okay. Constant thirst, additional effects, compromised ability to take up glucose, and increasing their appetite. Yeah, because they're not being able to take glucose in, so they're breaking their bodies down for energy, and so but they're not really because it's not like you're if you eat a meal, your body can break it down into glucose that you can use, then whatever, you're full. But if you know don't have the ability to do that and your energy is just from breaking down stuff in your body, then you know you're likely to be hungry because you're not getting the glucose from what you eat, I would say.
Hmm.
So yeah. So this would be like, you know, kind of like an inference type of question just because yeah, they didn't mention anything explicitly for this and the other choices are worse.
Okay. Yeah, this one I was a little confused about.
Mhm.
Certain bacterium was cultured for several generations. In a medium containing 15N transferred to a medium containing 14N was allowed to complete 2 rounds of cell division. Given that the bacterium's genome mass is 5.4 and grown in 14 and 5.5 and grown in 15, what is the um individual bacteria with which the following genome masses would be isolated from this culture?
Okay, so what are your thoughts here?
I mean, I had a feeling it'd be 5.4, but I wasn't sure. Like, should it be any of the others?
So what I would do here is, and like, I kind of realized this like maybe like a few weeks ago, but when I would be studying for the test or taking the test or practice test, I would actually write or draw things like all the time to help me visualize it. So, so because I don't know, like, so basically, so if I were to visualize this, right? So we have some something full of N15, and we incubate or culture, this bacterium. So it's going to be incorporating that N15 into itself. And then we transfer it to something that has N14, and it was able to complete 2 rounds of cell division. So it'll have, well, I guess if we have like one, it'll turn into the 2, which will turn into like the 4, which would have the N14 in it. And then it says, given that the bacterium's genome mass is 5.4 Fg. Oh yeah, what is Fg, by the
No
way?
idea.
So, I guess, yeah, when I talk about like the different prefixes, I usually stop at pico,
Yeah.
but I guess I was wrong in stopping there because this was a practice test. So yeah, there's gonna be, so like after pico, which is 10 to the negative 12, it's going to be femto, which will be 10 to the minus 15. So I guess, yeah, from now on, I would go by up to the power of 18 or negative 18. Um, that would be atto, by the way,
hmm
to the negative 18. So, all right. So yeah, that's what femto is. I mean, we don't really need to know it for this to answer this, but yeah. So okay, genome's mass. Okay, so all right, it's 5.4 when it's grown in this oh wait, when it grows in the N14 medium, and 5.5, so I'll just say 5.4, 5.5 when grown in the N15 medium. Okay. Individual bacteria with which of the following genome masses would be likely to be isolated from this culture. So since we have like, so this first one would be completely like 5.5, and then it'll undergo like the cell division, which will then make it. So essentially, this is just going to be a mix of the two, right?
So is the whole point that like when it goes through the first cell division, whatever, it's 5.45, and then the second cell division, it's 5.4. Is that like what they're getting at?
Yeah,
Okay.
yeah. So I mean, since we know it must be a mix, I would eliminate A and D,
Okay, got it.
and yeah, and then C looks very good to me, but we know that when it was in the N15 culture, it was for several generations. And when it's in the N14 one, it was like you know, like it's like two generations. But let's see. Oh yeah. So okay, yeah. So I forgot that we're talking about DNA here, so that would be easier. So I don't know if we did like a question before about like something where we have some DNA strand and then we know it's going to, let's say we put it in a new medium.
Yeah.
Yeah, it's going to be like this, semi-conservative. So let's say that the green here is like, the N14. And the blue here is the N15. And then, this would unravel to give us this. And now we don't have any more of the N15 ones available. So now, this next one will have to be both N14, right?
Yeah.
So initially I liked C because it's right in the middle. And, that's good. But now I see from doing this that we're gonna have more of the green, more of the N14 than we do the N15.
Yeah.
Right? Because yeah, once it starts, so we essentially have like a much larger amount of the um N14 one.
Mhm.
So now I would say so C would be if there were an equal amount, but now I would go for B because we know that there's going to be more of the N14, more of the lighter one.
Okay.
You want me to fear what I wrote or?
Uh, yeah. Okay. All right.
Mhm.
Uh, what's your usual drink of choice? Like when you're drinking that cup, is it like tea, coffee,
Oh,
or like,
just coffee.
uh-huh.
Um, yeah. What's your usual?
Well, honestly, these days I'm either drinking water or like I used to drink like energy drinks, like not often. I'm like very careful with my caffeine intake, but like every so often I actually end up taking like half a caffeine pill. I don't know if you ever tried caffeine pills.
What's the strength?
Well, it's 100 mg pill, but like I split it. So I take 50 milligrams sometimes.
Mhm.
But it's like you have to take it in the morning or else because it's more of like a steadily effect as opposed to any drinks. Like a lot of the time you crash. So yeah,
Uh-huh.
I was just wondering. Yeah, it's just easy to get. Like it's easy to just start not abusing caffeine, but just like getting used to it, I guess the effects.
Uh-huh.
So I try to drink it or like take like the pill or whatever, like every other day. I don't know, not every day. I'm like very careful.
Uh, yeah, yeah. I drink a lot, and like
Uh,
over time, like, well, I guess like there was a time where I didn't really drink much of it, and then
uh-huh.
was a time when I drank a lot, mostly like when I started to have like
Yeah,
work hours, but
yeah.
there was also a time where I would just be drinking like Bang, like energy drinks.
Right. Yep.
So,
You're
uh,
able to sleep fine though. Like it doesn't affect your circadian rhythm, like when you do drink it later, you know?
well, my circadian rhythm's always been bad. Like
Really.
I've been taking sleep medications for like
Mm-hmm.
almost all my, well, yeah, like almost all my life. So I
Right.
had like delayed sleep kind of onset disorder.
Mm,
You just kind of naturally fall asleep at a later time.
okay.
Do you have something similar? Because sometimes, you message me and it's like,
Yeah, it's
hey, are you like also awake?
the thing is, yeah, I mean, my sleep schedule's been all over the place with like the studying. Like, because the thing is also the days that I take a caffeine pill, like I, it's hard for me to fall asleep. Like also,
Yeah.
like I stopped doing it. I take it a lot less. At the beginning of the not semester, the summer, like I would be taking 200 milligrams of a caffeine pill.
Uh-huh.
Um, and I'm going from like drinking like a sugar-free 80 milligram Red Bull to taking a 200 mg caffeine pill, and that like, yeah. But now that I go back to college, I'm gonna have to completely change around because also I'm more productive in the mornings.
Yeah.
And during the summer, I have not been waking up too early. Like I stay up late and work up late. So,
Yeah. 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 It
but
definitely gets
yeah,
bad when it's like when it's like winter and the sun sets at like 4 p.m. and I'm like,
yep, makes sense.
but yeah, okay.
Okay. Um, before like we do the passage base, I want to quickly get this one out of the way. So yeah, showed a reaction and it basically asked how can we like track it, I guess. And determining the reactant, which reactant loses the OH group, which of the following isotopic substitutions would be most useful. So, I thought that here, like, we would replace the yeah, I think I put A, that we would replace the hydrogen that's on the acetic acid.
Um,
So
so what would happen in this reaction, by the way? Like,
the alcohol would attack the carbonyl, and
yeah,
then yeah.
good. Now, for the acetic acid, um, it's going to want to get rid of that proton, right?
Yeah.
Like whether the ethanol is there or not, right? It's just gonna get rid of it.
Okay.
So if you labeled that acidic H, it's gonna be lost like right away.
Mm, yeah.
So, but you know the mechanism, which is like 90% of it, like good job on that. So, if this attacks, right, this goes up, makes that intermediate, eventually this goes back down, and then this leaves as a leaving group, right?
Hmm.
So then we get this, right?
Yeah.
So so in order for us to figure out if this reaction's like working in this mechanistic way, we would have to see if we either are adding this oxygen from that ethanol. So we could label that. So I'm going to just make that green. Or we can label, I guess, this one. Because this is one that we removed, but it's going to be taken up by any of like a billion whatever molecules of water, so it's not going to necessarily show up on this molecule of water.
Yeah.
So basically, if I give you a beaker of acetic acid and a beaker of ethanol, and you label the ethanol and we mix them and we get the label for ethanol, like the oxygen of the ethanol, then we know that the reaction worked.
Yeah.
So that's how we know mechanisms. There's no other way for us to know in real life, right? Like, because I always think like, okay, how do we know it's doing that, right? With the arrows and all that stuff.
Yeah.
It's through the isotopic labeling. So yeah, we don't want this because that is just going to leave. But we want to see what changes, and what changes is we are taking the oxygen of the ethanol and adding it to the acetic acid. The carbonyl oxygen doesn't change. It's the same one. So it should be the hydroxyl oxygen, right?
Yeah. Okay.
Yeah. I guess like try to like I don't know, like what helps me all the time is just visualizing it. So if you can visualize it, I think that will help.
Okay. Uh, yeah. Yeah, I'm just looking at what I got wrong.
Mhm.
Uh, yeah, could do this real quick. So we got two questions wrong here. In terms of time, might just be good to just go over it right off the bat, maybe. Or I could quickly read through it.
Um, I can quickly read through it.
Sure.
Um, yeah, they're trying to figure out the amino acids of primitive Earth. Okay, malic acid is thought to have formed via hydrolysis, buzzword right there, of a cyanohydrin intermediate. We're not expected to know what that looks like, so they're going to give it to us. Compound, and then I'll take a look at the scheme. So compound A, we have what I would assume to be malic acid, and we are adding cyanide to it, and it gets added and turns into the cyanohydrin intermediate over here. And of course, it's going to be doing that, right? So, all right, then we add water to it, the intermediate here, and it seems to kick out this. That's a little annoying. Well, actually, never mind. That is cyanide. Okay. So, yeah. So, okay, it seems to kick out that cyanide and produce this acid here. Oh, crap. That's malic acid, not compound A. Anyway, cool. So, and then it turns into this fumaric maleic acid. And it seems like what's happening here is, well, it should be, I think it's getting reduced alcohol to form the double bond. So, okay. Subsequent dehydration of malic acid results in the formation of fumaric acid. Okay, so dehydration over here. So water likely comes out. So, okay. And they give us the melting point, sure. And it's cis isomer maleic acid and the melting point of that. And then ammonia was thought to be a constituent of the early atmosphere, and it was proposed that it reacted with fumaric maleic and malic acids to afford a pre-biological synthesis of aspartic acid and amino acid. Oh, I see. So that first scheme was like how an acid might have formed, and now to make an amino acid, we would want to add, nitrogen to it, and we can do that by adding, ammonia. So okay, so we see in the scheme, malic acid plus ammonia gives you aspartic acid, and then oh, and then this was like the fumaric acid that also, if you give it ammonia, turns into the aspartic acid. So okay, in an attempt to verify this hypothesis, chemists tried to reproduce the synthesis of aspartic acid under simulated primitive Earth conditions. An open flask containing malic acid was treated with a continuous ammonia feed, and the mixture was heated at 150 C, which is between these two.
Yep.
For 1.5 hours, the experiment was repeated with a mixture of fumaric and maleic acid. So instead of just the malic acid one, they did it with this mixture here. The results were analyzed by paper chromatography. The RF values were compared to the of the mixture was compared to that of the aspartic acid, blah blah blah. So, all right, cool. Okay, what assumption is being made if scientists conclude that aspartic acid was formed by the pre-biological synthesis in the passage? Okay. Now, so if the scientists are saying that, all of this stuff is what actually happened, what assumption are they making? So we can just like look at the choices. So, yeah, what do you think about these choices?
You potentially see.
What was that?
Maybe that they were like available in the first place.
Uh-huh. So when you say, tell me what do you mean when you say that?
Like C, compound A, and cyanide. Well, actually, hold on. So we can go through them from A to D. A, aspartic acid is stable, unstable at temperatures below 150. So I wasn't sure about that answer. I was tripped up by it on the exam.
Uh-huh. So, let's say that this is an assumption that the scientists are made. Um, uh, well, they say here that whatever, they heated the mixture up to 150 C. But they're not really saying that they're heating it up because otherwise aspartic acid would break down, right? So this is not an assumption that we need to make in order to you know prove this hypothesis, right? Because the only thing that they're saying is that they heated it up to 150 C, but they're not doing that because it's because it's unstable at lower temperatures. So don't think it would be that. Um,
B, I mean, they said all, so I was kind of like, no.
uh-huh. Yeah. You don't let yeah, you want to be careful of extreme words like that. But if it is true, um, well, yeah. So yeah, I'll go with you on that. Like, it's not. It doesn't have to be all of it. To form because you could still have some of this. And I guess here it kind of actually shows you that you could still have some malic acid to form the aspartic acid, right? So if all of the malic acid turned into fumaric and maleic acid, we should only see this part, right? There wouldn't be any of this left.
Okay. See, so they spoke about compound A and cyanide.
Yep.
I feel like it could be it.
Yeah, because in this primitive Earth, right? All of this stuff would have to exist, right?
Yeah.
And then I guess so D says reaction between ammonia and fumaric acid was catalyzed by the presence of water, but I don't see that. Like they're saying this to this.
Yeah, like it wasn't mentioned. Is that what you're saying?
Yeah. And like, if it, I mean, if it was catalyzed by the presence of water, that should be like a hydrolysis type of reaction, which is what we don't see. So yeah, it should be C. The assumption is like if they're saying that this is how amino acids formed on primitive Earth, they are making the assumption that like the things that they're saying made the amino acids like have to exist on have to have existed on primitive Earth.
Mhm.
Yeah.
All right. So, which of the following statements does not correctly describe the dehydration of malic acid to form fumaric and maleic acid? Yeah. So, reaction occurs most readily with tertiary alcohols. Okay. I think I put that. So,
So
yeah.
uh so what is this is like kind of like so what do they I guess mean when they say tertiary alcohols or something?
I mean, it's attached to 3 carbons. Like, I mean, it's attached to a carbon that's attached to 3 carbons, right?
Uh-huh. And so this is kind of like an SN1 SN2 type of question in disguise, at least some of the choices. Like because like what reaction would most readily react with tertiary alcohols? Like that would be like SN2 stuff. Or sorry, yeah,
SN1, right?
S SN1, yeah.
Yeah.
Or steric hindrance SN1, yeah. So this would be like, yeah, SN1. I mean, possibly E1 as well. Um, this is the dehydration thing that they're showing us. Um, okay. Then, all right, involves a loss of a water molecule, which is what we would have, right? They didn't draw it, but they did say that it was a dehydration, right?
Yep.
So this is true. And we're looking for not, so that shouldn't be it. Now, C is also one of the SN1, SN2 stuff in disguise, right?
Yeah. It's SN1, right? So carbocation intermediate.
Uh-huh. So this would be consistent with A. So that shouldn't be it. And and then stereospecific, that's also an SN1, SN2 thing in disguise. Because SN one, right, would produce that racemic mixture
Mhm.
that, is not stereo specific, but SN2 would be, when you have that inversion of stereochemistry.
So when something is stereo specific, does that mean like it's talking about SN2?
Uh, well, I guess it just means that the stereochemistry matters. Um, like, so like a lot of times that could be like, some type of enzyme can only work for a certain type of like of enantiomer, um, and like not the other type of enantiomer. And so yeah, with SN1, you can have that nucleophile attack from either side of that carbocation intermediate to make an equal amount of both enantiomers, but which would mean that it's not stereospecific. But in SN2, you're going to get that inversion. So we know which is stereospecific. So that uh, isn't going to be what we see there.
Question.
Okay.
Okay, yeah. Initial filtration step in the glomerulus of the mammalian kidney occurs primarily by A, passive flow to a pressure difference, passive flow resulting from a countercurrent exchange system, active transport of water followed by movement of electrolytes from resulting concentration gradient, and then active transport of electrolytes followed by passive flow of water along the resulting osmolarity gradient. Uh, I mean, I just got tricked by this. So it was like, I put B, I saw countercurrent exchange. I just like kept going.
Uh-huh.
But yeah,
Got it. So, um, it's okay. So we talked a bit about the passive flow versus active transport. Uh, so you are aware of that now. And oh, and you said from the counter current exchange.
yeah.
So, uh, what is that? What is the counter current exchange?
All I remember is that it's like the vasa recta and the loop of Henle are they're like connected.
Yeah, so like that entire nephron tube system, like all throughout it, like usually it's not drawn that way because it's hard to draw it, but all throughout that, like surrounding it and stuff, are a bunch of like capillaries that produce that vasa recta. So if things leave the tubes, uh they go into the vasa recta, and we call that reabsorption because it goes back into the bloodstream.
Mhm.
So um so that would be like a good choice for most of the nephron, but what's that initial filtration step um in the glomerulus?
I don't really know. I mean, I think yeah, I don't know. I don't remember. I just don't remember if it's big or small things.
Uh where do you think it'll be here?
Very left.
Show me.
Do you see the arrow?
So there, right? Uh-huh. Yep. So so uh so this is the start of like this tube system, right?
Yep.
Bowman's capsule.
Yeah.
What's this?
Glomerulus,
Which is?
capillaries.
Uh-huh. So how do you know, like, so we're essentially filtering our blood by getting it from the glomerulus into the Bowman's capsule. And how does that work?
I don't remember. Is it like small things that get filtered out or big ones?
Oh, well, yeah, small things and stuff like that. But like throughout the nephron, there's like this, you know, solute concentration goes up if you go.
That, like down, yeah.
So you'll see throughout the nephron like things happening because of that osmolarity.
Right.
But here, we're not having an osmolarity difference here. So the only reason that it's going from the glomerulus into the Bowman's capsule is because of hydrostatic pressure, in this case, which is blood pressure. So it's just that the pressure is high at the glomerulus that it forces it through. Yeah.
Mhm.
Well, when I say forces it through, it makes it sound like it's active, but it's going to be passive flow. But so you had the right idea, um, in terms of passive flow. And if they mentioned anything else in the glomerulus, you would be right for the countercurrent exchange part. Um, well, I guess except for the parts that are active transport, but yeah, it's
Yeah.
passive because of the pressure difference.
But when is it active transport? Like what is that? What like when you were like the ATP, NAK, ATPases?
So
Like
whenever you have yeah, so it'll be those types of things, but it'll happen whenever you have uh things moving against their concentration gradient.
but where is that in the kidney? Not from.
So that would be, for instance, um like for the ascending loop of Henle, you will have um sodium getting reabsorbed. And so, like, for instance, like, okay, this descending loop enters into a, hypertonic environment. So water is going to be sucked out uh, through passive transport.
Mhm.
In this ascending loop of Henle, we can have salts like NaCl come out. Now, this is a high solute concentration region. So the salts should not be coming into that high solute concentration region, right? So
Yeah.
it goes against its concentration gradient, and that is an example of uh, active transport here. Um, and you can also have that happen like in some other regions, like in the distal convoluted tubule, you can have that happen. You can have that happen in the collecting duct as well. So, uh, so yeah, I would say you'll have some of it happen in the proximal. You'll have none of it happen in the descending, and the ascending. Like if I drew this really kind of properly, like there would be like the thin part, and then the thick part of the loop of Henle. So uh at the thick part of the loop of Henle, you can have active transport happen. You could have some of it happen at the distal convoluted tubule, and then you can have some of it happen at the collecting duct. The ones that'll happen at the collecting duct will happen due to like something like aldosterone or something like that.
Yeah.
But yeah, that's um yeah, those would be the locations of where that kind of thing happens.
Okay.
Yeah, here's a
Uh,
picture in the chat. So
mhm,
you could,
yeah.
yeah, you could see that the red arrows, active transport, passive transport are like the whatever, I think purple arrows or whatever. So you can see that in the proximal tubule, there's some active transport happening. Descending loop, only diffusion or passive. Ascending loop, like thin portion is passive. Thick portion starts to become active, and then the distal convoluted tubule has a good amount of active transport happening, and then in the collecting duct you can have some active transport happening as well.
I just don't get why in NaCl on the thin part, like why is it going through passive transport if there's already a high solute concentration? So like there's more now solute coming in.
Uh-huh.
How's that passive, you know?
Oh yeah. Yeah, I guess.
So I guess gotta memorize it. You're against memorizing, but
Yeah.
yeah.
Yeah, I can yeah. I can look into that and get back to you.
Yeah. Okay. Got it.
Okay.
Okay. Sounds good. Um, what will be the total for today? Start off again.
What's up?
What will be the total for today?
Oh yeah. 113.
Okay. Well, I guess September. I'll see you then or
Oh yeah, yeah. Yeah. So like I was gonna make like an analogy about like running. Like if you don't have training running and then you just start sprinting, like compared to like if you slowly, kind of get up to that point. I think what we were doing with the UWorld problems were like going into like sprinting like without like solidifying the basics, and I think that like would just like make it really really kind of like make it so that it can like really like psych you out. So like in hindsight I think like we would have done better if we uh kind of like started from more of like the basics, but that's what we did today. So and you can see how these questions aren't like trying to kill you like the rolled ones. So yeah, I think that you know to yeah, so I think you're in a good place. And essentially yeah, just enjoy the rest of it. Yeah, just take a complete break from anything MCAT related. Um, get a chance to recuperate, enjoy the rest of your summer. Just like put yourself, in a different headspace, because I think that yeah, I just think that it was just a confidence thing. And I think that the really hard world stuff made it so like, if we did like the basics and all that stuff, and then we worked up to the world problems that were really tough, that would have been like better, but and you know, it's my, you know, it's my fault for this. Like I should have kind of stopped it at some point and just made sure that we went over, you know, more of the basic stuff. But I think that that could have played a role in like kind of like psyching you out when you saw these AMC tests, because maybe you were expecting it to be like of the difficulty level of the of U World. So maybe you like would choose, or maybe like it made you like more suspicious of, you know, maybe this is too straightforward.
You're right. Yeah. No, that totally makes sense. Like what you just said, yeah. Like there were times where I'm just like, there's no way it's that simple, you know?
Yeah. Yeah.
I was like that during the test for sure.
Yeah, because yeah, I never see you know, it's I usually see blueprint scores being deflated, like, you know, I rarely ever see that
Right.
the other way around. So I think
Yeah.
that yeah, so I think it's not like a real issue like on your part in terms of like knowledge or, you know, skill or ability or anything like that. I think it's just uh the headspace was kind of messed up by some of the stuff that we were doing. So so yeah, so just straight up don't do anything MCAT related. Don't even think about it. Don't work, like I know there could be some worry of, am I gonna forget stuff? Should I still be doing Anki? But you know, you're gonna have it's not it's not gonna be hard to like remember the stuff again. And a lot of times it's better to like have a little like break like that, so your brain gets a chance to kind of I guess crystallize the stuff that you know, and but also makes it more like flexible because you're not like constantly worrying about, or you know, doing work and stuff like that. So yeah, I would say just enjoy the rest of your summer. You know, how's your injury like
But
healed?
way better. That's gone way better. Only like the first 2 weeks
Yep.
were tough.
You're
So you're
probably
saying,
running.
yeah?
Yeah.
Not yet, but soon. I just hit like legs in the gym for the first time in 3 months yesterday. And it was like a breath of fresh air.
Nice. Yeah.
Yeah.
Like squats and stuff.
Slowly but surely. I was doing like the lowest weight, you know, but it was still like nice.
Yep. Awesome. Yeah. So yeah. Yeah, enjoy the rest of your summer, and then yeah, hit me up when you like yeah, when you want to.
Go back. Yeah.
And then we'll make a new schedule with like stuff like foundational stuff first and all that stuff.
Okay. Thank you so much, Shane.
Yeah. So yeah, enjoy.